Okay, so I'll start us off with a prayer. Dear Heavenly Father, we're grateful for today. We're grateful that we get to meet together, and we're grateful for the opportunities that we have to learn. Please help us so we can have Thy Spirit with us as we discuss how to culture microbes in the lab, and that the students can be able to understand the new concepts that we're learning, and feel free to ask questions, and that their studying will be effective. And we love Thee, and we say these things in the name of Jesus Christ. Amen. Okay, let me spare the chat box so I can watch that. Okay. Okay, so first off, sorry, new class, so new groups. So those will be posted, tomorrow night. So be watching for that. So tonight, if you haven't filled out the doodle poll and you want to, fill it out. Right now, your Unit 1 exam score does not reflect any extra credit points if you participated in that discussion board. And so if you did meet those requirements for that discussion board, you'll see your score go up a little bit by the end of the week. I wanted you to have a chance to kind of see your score before the extra credit changed it, to kind of give you an idea. So just a reminder, for the Unit 2 assessment, we will be using Proctario software. So if you haven't installed that software and done the practice thing in Chrome, make sure you do. You need to make sure you use the Chrome browser when you do Proctario. Once you have your new group assignments, try to get in touch with your group members by Saturday. You will have, you can email each other and use iLearn to contact each other the same way you did for the disease presentations. You will also have a discussion board available to you every week that we have a case study. We have 2 case studies this unit. So we have one in week 7 and one in week 9, and then we have 3 case studies of the 4 weeks in that unit. Unit 3. And so I would, I would recommend if you're trying to get in touch with your group members, make initial contact. If you use that discussion board, just keep in mind there is no discussion board available in Lesson 6 because there's not a case study, but you have access to everything in Lesson 7, so you can post there in that case study discussion board. And you should watch for that if you're trying to make contact with group members as well, to see if they have posted there. So make a plan. I would recommend this week that you make a plan for how you're going to work together, so that Monday you can get started on it right away. So you could meet synchronously in Zoom if you want. You each should have access to your Zoom rooms like you did, many of you did, for recording your presentations. There's Google Hangouts, there's Skype. You can also work on them asynchronously via the discussion boards, or lots of people use Google Docs to peer review. Make time to peer review. These assignments should not be, they are designed to be a little bit more difficult, so that you do kind of have to work together on them. The questions for the case studies show up on the exams, and so you are held accountable for everything that's on there. So I would not recommend the strategy of everyone take 2 questions and compile it all together, because if something is wrong, then you lose the points for that. Obviously, and then you don't know it for the exam. So do make time to peer review to make sure things are accurate, and try and work on them together. That's the goal, to teach one another. That's why we have these. It's a big goal for BYU Idaho, is that as we teach each other, we learn better. Just a reminder, no plagiarism in the case studies. So again, I would recommend that whoever submits it checks out their originality score and turn it in, and kind of looks to see if there are any highlighted areas. And just in case, even if you didn't, maybe a group member did advertently or inadvertently, and no quotes as well. Things do need to be in your own words. Part of that is because that being able to accurately summarize something shows that you've mastered that concept. Quotes just show me that you know how to Google. And just a reminder, stay away from things like Course Hero, Quizlet, and stuff like that. And also, you should not be sharing work with future students or whatever either. Okay. Just wanted to touch base on on one thing with the clinical, a couple things with the clinical submission. So the viral envelope, there were a few people who said the viral envelope is there to hold the spikes that let the virus get in. An envelope is not essential for that, right? Because non-enveloped viruses also have spikes that are responsible for helping the virus get in. But with an enveloped virus, the spikes have to be poked through the envelope or they'd never be able to make contact with the host cell, right? Some people talked about how it was helpful in allowing the virus to enter host cells, which is accurate because it is a piece of host cell membrane, and so it can kind of fuse together with that and get in better. The number one answer that we were looking for, though, was that that it provides that camouflage that makes it harder for the immune system to recognize the virus because that membrane is made out of host cell membrane. So, or that envelope is made out of host cell membrane. Jennifer, yeah, you can use Quizlet to create your own study guides. That's fine if you privatize them. Yeah, it's just in the past we've had issues with students posting actual exam and assessment questions and stuff like that, which they shouldn't have been doing. If you want to use it for your own studying, that's like totally fine. Just don't go looking for other people's stuff. So chickenpox versus shingles, some people said there was not a rash difference because both have rashes on the skin. It's true that they both have rashes on the skin, but the location of the rashes is usually different. And part of that has to do with where the virus is hanging out in the body. So chickenpox is contracted via that respiratory route and those respiratory secretions. And so when you contract chickenpox, it gets into your respiratory system, and then it circulates throughout your blood, throughout your whole body, and so that's why you typically see those lesions everywhere. With shingles, it's emerging from the nerve cells that it has been hanging out dormant in, that latency. And so when you see that shingles rash, it's typically on the skin that is directly above the nerves where it has been hanging out dormant. And so that's why if you have shingles more than once, oftentimes you see the rash in the same place because those are the nerve cells that it's hanging out in. So it's kind of what that answer was looking for. And so a lot of times with shingles, you'll see it on the trunk of the body, maybe oftentimes just on one side of the body. And that rash is more concentrated in that one area instead of kind of all over like chickenpox is. So disease presentations, you're going to be watching those. Those are posted for this week, so you can go check those out tonight or sometime this week. Make sure that you're filling out your spreadsheet. That spreadsheet will be helpful as you are preparing for the unit assessment or the lesson assessment, the unit assessment, and the final. It'll be nice to have all that information together. The other nice thing about the spreadsheet is it helps you know what to pay attention to. Make sure that you have the correct names. So for bacteria, we're looking for that binomial nomenclature, which is the genus and the species name. And then for virus, we're looking for the species name of the virus. Big hint, for most of the viral diseases that we're talking about, the species name of the virus is just the name of the disease. Mono is different. It's caused by the Epstein-Barr virus. But most of the other viruses that we talk about, it's like measles virus, just measles virus, Zika virus, respiratory syncytial virus this week is just respiratory syncytial virus. That's the species name. So don't get caught up in the more exotic sounding genus, family, order, whatever names. We want the species name. Anything higher is not specific enough. Watch for things like for viruses, what type of genome it has, if it's a DNA or a double-stranded RNA or single-stranded RNA. If it's a single-stranded RNA, is it positive sense or negative sense? Is it enveloped or non-enveloped? If it's a bacterium, is it gram-positive? Is it gram-negative? Does it have a unique shape or any special defining kind of characteristics or features? If there's a vaccine, what type is the vaccine? Is it a attenuated live vaccine, which means the microbe is active and can infect people, but it's weakened, which means that it doesn't cause illness. Is it a subunit vaccine or an acellular vaccine? Is it a what kind of vaccine is it? If it's bacterium, does it make any special toxins? Or other unique characteristics. For example, when you're talking about or studying leprosy, leprosy is what's called the mycobacterium. It has a extra special cell envelope. It's a little bit different than most other bacteria. Treatment and prevention, be paying attention to if there are medications or antibiotics that are an option, as well as supportive treatments. Those are also important. I would not worry so much about which antibiotics groups list as an option for treatment. Instead, just focus on whether antibiotics are an option or not. We'll talk a little bit more about antibiotics in lesson 9. That's when we'll focus on them. But the antibiotics that the groups picked are not necessarily ones that you need to remember. You should be writing down what the reservoir for the microbe is, signs and symptoms, those things that are in the study guide. And I will, sometimes the presentations are not 100% accurate, or maybe they're missing some important information, or there's just something that I want to emphasize extra. So also maybe watch for any addendums that I might add in our weekly meeting, and pay attention to those so that I'll try and catch any issues. So the Unit 1 assessment is graded. The high was a 97.5. We have 2 people who got that score. And the low is a 34, and the average was a 78%, which is good. So overall, you guys did a good job on that exam. I was impressed with that. If you want to know what you missed, email me and let me know. The easiest way initially usually is just for me to send you an email and say, these are the concepts that you missed and that you'll want to review. If that's not sufficient for you, then you can let me know and we can do something further, like meet in Zoom, or you call me on the phone or something like that. So just a couple of things I wanted to highlight for the disease presentations this week. So humans are the reservoir. I just kind of wanted to clarify. So sometimes there are bacteria that people can end up being long-term asymptomatic carriers for. If you've ever heard of Typhoid Mary, she is someone who was a asymptomatic carrier of Salmonella typhi, which causes typhoid. She had initially contracted it. The bacterium took up residence in her gallbladder, and she infected quite a few people. She was a cook and infected quite a few people that way. So pertussis is not that kind of disease. There are not people roaming around who are long-term carriers for pertussis. Are long-term asymptomatic carriers for pertussis for the most part. If you have it, you have symptoms. Sometimes you can be, if you have been vaccinated against it, you might come into contact with it and contract a little bit of an infection but not show any signs or symptoms, which is but that's not common, and it's not a long-term thing. Like that would be like you had it for a few days and then you cleared it out kind of thing. So we are the reservoirs, but again, it's not like it's hanging out a long time in your body. So the group did a really great job of explaining vaccine types. I just wanted to highlight that because it might be something that is new to you to kind of pay attention to. So pertussis is what is called an acellular vaccine. That means it contains pieces of the bacteria but not whole cells. So sometimes you have, like I said, that attenuated live vaccine, which the microbe can infect to a certain extent, but not enough to cause illness. Sometimes you take the whole microbe and you kill it and you inject that whole thing. This is just containing pieces of that. So kids get DTaP and adults get Tdap. And I was glad they talked about vaccinations for pregnant women because that is something that they started doing the last 5 or 6 years, because if the mother's vaccinated in that 3rd trimester, then the baby is born with some of those antibodies against that, which can help them those first couple of months before they can be vaccinated. I just did also want to mention as well, they talked about antibiotics as an option. Supportive treatments are also an important part of treating pertussis. So things like providing oxygen for people who are having trouble breathing, steroids, breathing treatments, a mechanical ventilator if necessary, or hospitalization in very severe cases. And if you've never, I would recommend, if you've never heard the pertussis cough, the whooping cough, to maybe look it up on Google a clip on YouTube or something, because it is a very distinctive cough and kind of interesting sound that pertussis has. So for RSV, it's respiratory syncytial virus, and again, just a reminder, use the species name for that, not the most technical sounding name. Okay. So today we get to talk about culturing microbes. So these are there was a journal that kind of had this nerdy, sciencey competition that asked people to make art with culturing microbes. And these are some of the things that they got. Some bacteria, so the picture on the right is most likely not bacteria, it's probably yeast, but some types of microbes have pigments. And as we talk about today, some media change color as microbes are grown on them. So these were kind of some of the entries that they got. And then the kind of classic, again, nerdy microbiology joke, bacteria, the only culture some people have. So we're going to talk about today. Okay, so one of the things that I want you to pay attention to today and this week, again, are the differences here between bacteria and viruses. And I want you to, the methods that we talk about today, be asking yourself, would I use these for bacteria or for viruses? So the question is, can we culture bacteria and viruses in the same way? Why or why not? So what do you think? Can we culture them the same way in the lab? Use the same methods, same techniques? So I have one no in the chat box, and that is correct. We cannot culture them in the same way. So most of the methods that we're going to talk about today for bacteria are, we provide bacteria food, some kind of food, and the environmental conditions that they like to grow in, and they grow. We know that viruses don't have any of their own machinery for metabolizing energy, making proteins, making lipids, replicating genomes. And so for viruses, we can't just give them supplies. We have to give them host cells in order to grow. And there is, I posted a link to a little article and kind of a couple of questions to think about this week also. With bacteria and how many of them we can actually culture in the lab. It's pretty small. Scientists estimate that we are really only able to culture about 1% of the bacteria that's out there. So if that's something that sounds interesting to you, I'd recommend checking it out. Okay, when we're culturing bacteria, we usually use one of two methods in the lab. We either use the solid agar or the liquid broth. And most of you have probably done something with this solid agar before, right? The classic, find something gross and swab it and rub it on the agar, or wash your hands and, stick your thumb on there and see what happens. And agar has some special compounds extracted from seaweed that allow it to be solid at warmer temperatures, room temperature, body temperature, whereas things like gelatin, if you tried to use that, it would liquefy at those warmer temperatures. So that's kind of something that's special about agar. It keeps your media solid at higher temperatures. And this liquid is a kind of a good example of what Pasteur had in his Swan-neck flasks when he did that experiment disproving abiogenesis. For viruses, like I said, we have to give them cells. So some of the kind of classic examples that we use are embryonated eggs we can use to try and culture viruses. Animals, We can culture viruses in animals and cell cultures. If you have ever read the book, The Immortal Life of Henrietta Lacks, that the cancer cells that were harvested from her are kind of one example of cells that you might use to try and culture viruses in that cell culture. So anyway, if you haven't read the book, I would recommend it. It's interesting. So this slide is for bacteria. Again, because viruses just require, their oxygen requirements are just for whatever the host cells that they grow and require. Some bacteria like oxygen, and some bacteria don't like oxygen. Some have to have it, and some are killed by it, right? And so if you have a bacterium and you're trying to figure out its oxygen requirements, you can perform this experiment where we have our tube, and within this tube is solid agar. And we get our bacteria on a wire. We usually call that an inoculating needle, and we stick it down all the way into this agar, and then we see where we have growth. So the cap on the top is fitted loosely, which means oxygen can diffuse, or gases can diffuse in and out, and some of that oxygen will diffuse down into the agar. If you, after incubating it, if you see growth only at the top, that tells you that your bacterium is an obligate aerobe. That means it has to have oxygen in order to grow and survive. If it doesn't have oxygen, it will die. If you only see growth at the bottom, that tells you it is an obligate anaerobe, kind of like the Clostridium tetani that we talked about in our clinical submission a couple of weeks ago. It's only growing, the oxygen doesn't diffuse all the way down into the tube, and so you're only seeing the growth at the bottom portion of that tube that is anoxic. Facultative anaerobes don't have to have oxygen in order to grow, but they grow better in the presence of it. So that's why on this third tube you see more growth at the top and some sparse growth at the bottom. Not as much, because again, it will grow better with oxygen, but it doesn't have to have it. And then this last option is what we call aerotolerant, where it doesn't really anaerobe. The oxygen's not toxic to it, but it doesn't really use it, so it doesn't. So it kind of, you see the same growth throughout the tube. It doesn't matter. So in order to culture a microbe in the lab, we have to provide it with its correct oxygen requirements. So we're going to be classifying media this week, and this is something that is sometimes difficult for students. So the first thing that when we talk about media, and again, when we're talking about media, we're just talking about bacteria, right? Because for viruses, we have to provide cells. We can either grow the bacteria in media that is in a solid state or a liquid state. And then we talk about the chemical composition of it. There are two categories here that we'll talk about. One is complex, one is defined, and then there are some other kind of things that we can tweak and change. And so to tell us different things about the microbes. So we also classify media kind of based on its purpose or what we're trying to learn from the microbe and using it. And so there are some terms we're going to talk about today that are selective, differential, enriched, reducing, etc., things like that. So the first thing that we're going to talk about is chemically defined versus complex. So this recipe on the left is what we call chemically defined media. If you look at this recipe, it is very detailed. It has very specific amounts of each of the components that we've added, and we know the chemical formula of each of those items. And so we know exactly how much cysteine is in there, and exactly how much thiamine, and exactly how much aspartic acid, and how much valine and threonine, how much iron chloride we've added, all of these things. We know the chemical formulas for all of them, and we know the exact amounts. We know everything that's in this media, exactly. The media on the right is what we call complex. And complex media, we don't know everything with the same level of detail that we do for chemically defined. And this is because we're adding some type of nutritional source that has a lot of these things in it, but we don't know the exact amount, like the exact amounts and exact breakdowns and things like that. And so when you are trying to decide if media is complex or chemically defined, you should look at the ingredients list. And anytime you see any animal parts, any names of organs, anything that says extract, or infusion or whatever, that tells you that it's complex. So this brain heart infusion broth, we have 27.5 grams of brain, of 25.7 grams of heart extract and peptone extract. So all of those things are going to have the nutrients that our microbe needs to grow. It's going to have amino acids, it's going to have salts, it's going to have vitamins, it's going to have sugars or fats or whatever, but we don't know the exact amounts the same way. So yeah, and I mean, and you might, so Jennifer says, so with complex, you don't know the ratio of the ingredients, but together they equal the 27.5. So that's, you know, the amount that you added, but I mean, there could be a little bit of, you know, a small amount of variation between when, you know, one set of brains that they added could have had a little bit more arginine than another set or something, but that you're not really like worried about that the same way. It's similar enough that it's okay. And when you use defined media versus the complex media really just kind of depends on your purpose and your experiment, or if you're trying to just grow it in the lab for there are some experiments where you might not need to know that level of detail or be worried about it, and you just need to grow it in your lab. And so the complex media, most of what you'll do if you were to take a lab class or work in a hospital or something like that, most of what you do would involve complex media. It's even though you don't know the level of detail that you do for chemically defined, you know enough and it's fine. Chemically defined media is a pain in the butt to make also. I've done it and it's a lot of work. Adding all these teeny tiny amounts of everything. You should be able to, if you were given a type of media and you had to research and you looked up the ingredients in it, or you're giving a list of ingredients or something like that, you should be able to recognize whether it's chemically defined or whether it's complex. And we'll have some practice questions on that later. So selective media is going to allow for the growth of certain microbes while purposely inhibiting the growth of others. One thing that you often add to media to make it selective are antibiotics. If you were trying to select for microbes that were resistant to a certain antibiotic and select against microbes that are susceptible to it. You might also add, so these are, this table is from your book and it has different types of media and an agent that they've added to make it be selective. And kind of what you'd use it for. So salt is another thing that we can add to a media, and microbes that are a little more tolerant of higher levels of salt will grow. Halotolerant or halophiles will grow, whereas microbes that are more sensitive to higher salt concentrations won't grow. pH is another one that we can do, whereas that can allow you to select for certain microbes and against others. And so this can be helpful in identifying microbes, and also if you're collecting a specimen from someone, oftentimes you have an idea of kind of what you're looking for, and you don't want to grow a bunch of stuff that you're not interested in, because we know that we have a whole ton of different microbes growing all over us. And so if you use selective media, you can kind of screen out a lot of the stuff that you're not interested in, and more kind of target in on what you're looking for. If you can find a selective agent that will select against certain things and for what you want. Differential media is another thing that we can use. So both selective and differential kind of fall under what I called that purpose category. So with differential media, certain microbes that grow on that media will look different than other microbes that grow on it. So blood agar is an example of a media that we can sometimes use in a differential fashion. Some bacteria will be able to lyse open red blood cells. We call that hemolysis, and some will not. So this, the little alpha, some bacteria can do what's called alpha hemolysis, which is partial lysis of red blood cells. That beta hemolysis is going to be that complete lysis of red blood cells, which is why you see where the bacteria have grown. You don't see the red from those the sheep's blood anymore, because all those red blood cells have been lysed open, and then that gamma hemolysis is where they're not being lysed open. And so if we are growing something on blood agar and we are looking for hemolysis activity, then the blood agar is acting as differential media. Again, because bacteria that can lyse open red blood cells look different when they're grown on it than bacteria that cannot. This is another example of differential media where bacteria that can ferment lactose. We've added lactose to this media, and we've added, it's called MacConkey agar, and we've added an indicator dye. And the indicator dye is going to change color based on the pH of the media. So if the bacterium on here is able to ferment that lactose, the fermentation byproducts that are created are going to drop the pH, and the dye is going to change color. So for the bacterium that is grown on the left, the bacteria still grows there. It's still growing on the media, but it doesn't ferment lactose, and so you don't see that color change. Whereas the middle and the bacterium on the far right, they both ferment lactose, so you get this change in pH, which causes a change in color because of the dye that you've added. So if you're trying to decide if media is differential or not, look for any clues that say that some microbes look different than others when being grown on the media. So that's kind of the key that you're going to want to be looking for there. And similar kind of thing to selective media, this can be a way for us to identify certain microbes that we might be looking for. Other types of media that we can use, enriched media. So sometimes you might need to add additional components to grow things that are a little bit more maybe finicky. When I was an undergrad, I grew, at BYU, I grew all the bacteria for the lab classes in the microbiology department. And there were some we had to grow on what's called chocolate agar, which is you've added the blood, but you add it when the agar is hot, and so it breaks open the red blood cells so they look brown. Neisseria gonorrhoeae was one that was a little more picky to grow, and so you had to grow it on the chocolate agar. It wouldn't grow on the sheep's blood agar. So enriched media is when we're adding extra things to try and help the growth of maybe more finicky stuff. Reducing media is used to grow anaerobic bacteria. So if you wanted to grow Clostridium tetani or Clostridium botulinum in the lab, you would need to use reducing media. So again, and we'll have some practices, but when you're determining what type of media you are dealing with, ask yourself the question, what is my goal, or what are the clues that this scenario is giving me in terms of what type of information is it giving me in terms of trying to figure out what kind of media you're dealing with. Another thing that is important for growing bacteria is temperature. So we have psychrophiles, which are cold-loving microbes, psychotrophs, which are kind of in-betweenies, mesophiles, like medium temperatures, right around human body temperature, thermophiles like it hot, and hypothermophiles, like that Thermus aquaticus they pulled from Yellowstone, that lets us do PCR, or any of those microbes that you find in the geysers and the hot pots there. Those are hypothermophiles, and they like it super, super, super hot. So what temperature classification do you think most pathogens are going to fall under? Psychrophile, mesophile, thermophile, hyperthermophile? So they're going to fall under that mesophile category because that if you look, it peaks right around human body temperature, which, it's not a surprise if they're pathogens. They've evolved to, so yes, degrees Celsius is what this is listed. If they're pathogens, they've evolved to infect us, and so they're going to like our temperature, right? So that's kind of the temperature, which you're going to see most of the pathogens. So kind of something to take note is a bacterium. You can kind of see that sometimes you have a little bit of overlap. So under the curve are the temperatures they're the temperatures that a microbe in that category could probably grow at. The peak is the optimal temperature for the microbe. So there are some microbes that, in the fridge, your food doesn't stay good forever, right? Eventually microbes get to it and it goes south, it goes bad. But it stays good for way longer than if you left it on the counter. And that's because when you stick it in the fridge, you've slowed down that rate of growth. But it's still there. And so when we're trying to determine whether a bacterium is a psychrophile, mesophile, thermophile, hypothermophile, we're looking at their optimal growth conditions. Not can they grow when it's cold or when it's hotter, but what temperature do they grow at best? Again, which is why most pathogens are going to be mesophiles, because they like to grow in us and they grow best at, right around our temperature, which is so anyway. So you guys are really lucky, because when I taught this class at BYU a long time ago, we made students do math for this. You don't have to do any math. You don't have any math equations, but you do need to know the different phases of growth in a bacterial growth curve. So this is an example of what would happen if we took some bacteria and we stuck it in a flask of liquid broth and we let it go for 24 hours, 48 hours. And kind of to see what happens. So when you first inoculate your culture, you're going to have what is called kind of a lag phase. And in that lag phase, the bacteria are, it's kind of a little bit of a shock to them. It's a new environment. It's maybe different than where they were before. And so there's going to be a little bit of time for them to adjust and prepare to start growing. And once they leave that lag phase, they're going to enter what is called the log or exponential phase. And so once they enter into this, this x-axis is time and the y-axis is the log number of cells. And so we see they enter this log or exponential phase, so that means that they're growing at a constant rate. They're constantly dividing. So maybe if it's E. coli and it's in optimal growth conditions, this could be every 16 or 18 minutes, the E. coli one bacterium divides into two. And then 60 minutes later, those two become 4, and then 60 minutes later, those 4 become 8, 16, and so on and so on. And so they're going to be in this for a while. And you can figure out that actual growth rate by measuring the slope of this line. And eventually, they're going to start to kind of run out of space. It's going to get a little squishy and squeezy in that culture, and there might not be as many nutrients available, and they're going to slow down. They're going to say, whoa, I can't keep growing at this rate. And they're going to enter what is called stationary phase. So if you were to look at most microbes in the world at this moment, most of them are going to be in stationary phase, right? Which is good because, if E. coli stayed in exponential phase indefinitely, it would, take over the world pretty fast. So stationary phase means that the rate of cell death is equal to the rate of cell growth. So even though you still have cells that are dividing, you also have some that are dying off, and so your actual population of bacteria doesn't change. And it'll hang out in the stationary phase for a while, but eventually food is going to start to become scarce and waste products are going to start to build up. And then you're going to enter this death or decline phase, and the bacteria are going to start dying faster than they're dividing, and your number of living bacteria is going to drop. If you look at your culture, it will probably look pretty cloudy still, right? And that's because the bacteria that are dead their parts are still in there, right? And so they're kind of taking up space still, but they're not alive and growing anymore. So I would be familiar with the name of each of these phases, as well as kind of what's happening at each phase. So lag phase, you don't really have growth yet. It's kind of getting adjusted. Log or exponential phase, you have that steady growth of division. Stationary phase, cell growth equals cell death. And then that death or decline phase, you have more death than life. So did Henrietta Lacks cells never reach a phase past exponential growth? So Henrietta Lacks, so that, I mean, that's kind of one of the issues with cancer. She had cervical cancer, a super, super crazy aggressive case of cervical cancer, just huge tumors that grew everywhere. It's pretty bad. So that's kind of one of the problems with cancer cells in general, is they don't recognize when they need to stop growing, and they just kind of take over. So it's a little different than the bacterial growth curve, but kind of, I mean, our human cells, right, have hit points where we have more of a stationary phase, right? As adults, we don't keep growing, but cancer cells are a problem like that. That they just keep going, and they take up all the space, take up all the food, and they don't read those cues that the body is saying stop growing. But it's probably also one reason why her cancer cells, we were able to culture them in the lab when we'd had trouble growing others before, was because maybe they weren't so sensitive to whether the environment was perfect or not, right? Okay, so for bacteria, if we're trying to figure out how many bacteria we have, we can either use direct counting methods, or we can use indirect counting methods. So if we're using a direct counting method, we're actually physically counting bacterial cells. So we can do this through two different ways. We can do plate counts, or we can do it through microscopy. So for microscopy, we have this slide called a cytometer, and it's a special slide that has microscopic grids etched into the slide. And you add some liquid culture that contains bacteria onto your slide, and you pick a certain number of grids that you're going to count. And you know how much liquid those grids can hold, and so you count how many cells are on it, and then you use a little math equation, and it will tell you how many bacteria per microliter or per mil you have in your culture. We can also use plate counts, which is this cute picture down here, where we take a known amount of liquid culture and we squirt it on our plate, and we spread it around, and we let it grow, and then we count how many colonies we can see in these little spots, these little dots that we see here. These are colonies. And as long as you didn't add too many bacteria to your plate, each colony should represent one single bacterial cell initially. And then, it entered that exponential growth phase, and it grew over a day or two into a colony that you can see. So if I added 100 microliters to that slide, and I spread it around, and now I have 50 colonies on my plate, that tells me I have 50 bacteria per 100 microliters. And so again, for both of these, we're actually counting bacterial cells. Direct counting methods are actually counting bacterial cells. We can also use indirect methods to get an idea of how many bacteria we're dealing with. And kind of the three, and so our keyword for tonight is turbidity, if you want to write it down. So 3 of the most common ways that we can do that are 1 through dry cell weight. So if we took our liquid culture and we stuck it in a centrifuge, it's going to spin really fast, and it's going to pull all our bacterial cells down to the bottom of our tube. And we're going to dump off the liquid, and we're going to dry out that gunk that's left at the bottom, that's all of our bacteria. We're going to dry it and weigh it. and probably again use some math equation, some math standard equation to take it back to bacteria per mil or whatever. But obviously the more dry weight we have, the more bacteria we had in our culture. Another thing that we can use is turbidity. So looking at how cloudy or how clear our liquid culture is. So we would take our sample that has our bacteria in it, and we'd put it in a little machine, the spectrometer that's going to shine light through the culture. So you can see here, if the culture is relatively clear, most of the light that we shine into it is going to get through the culture and to the other side where that light sensor is. But if there's a lot of bacterial growth like this vial on the right, most of that light is going to get blocked, and when we try and shine it through the culture, it's not going to hit that light sensor. And so the amount of light that we can shine through the culture can give us an idea of how many bacteria we have growing in it. And again, for all of these indirect methods, you're going to have some type of, you would have made some type of standard curve, and you'll plug it into a math equation that will tell you, well, if it's this cloudy, it has about this much bacteria in it, and if it's this cloudy, it has this much. A third thing that we can do is you look at the metabolic activity. And so a lot of times this will be similar to the differential media, where we have a bacterium that can do something special, and we kind of measure that. So we could, again, add lactose in some type of indicator dye, and we could use a light source again like this to determine how much color change we see. And so if you had added lactose and you were dealing with lactose fermenters, the pinker your media was, the more bacteria you had, because that meant you saw more metabolic activity if the color was darker. It was stronger, more intense, than if it was less intense. So for viruses, I want you to notice that for direct counting methods, we have no direct counting methods listed for viruses. Anyone have any ideas why that is? There's a couple of kind of limitations going on here. So first one, so you're not going to be able to use agar, right, and do these plate counts, because they can't grow on agar. They need cells. They need host cells. And the other one that Xenia listed is also correct. They're too small, so we can't use our microscopy. We can't use our light microscope to count it. Electron microscopes are great for a lot of things. They're not, because of the sample preparation, they're not great for counting, because you lose specimen as you're preparing it. And so we don't typically use it. We don't use it to count. It's really not as accurate, very accurate. So there are no direct methods for counting viruses because of those two limitations, size and requiring a host. So if we want to try and figure out how many viruses we have, we can do what are called cell plaques. So we add our virus to some type of cell culture, and then we look and see what happens to that cell culture. So we added there's a virus here, and it killed a whole bunch of our cells. And so the area, using those cell plaques, the area of clear space can tell us how many viruses we had, can give us an idea of whether we had a lot or not very much. So this is the only method that we're going to talk about for counting viruses, is these cell plaques. Okay, so here are some practice questions. So here is a media recipe, and I want you to tell me if this media is complex or chemically defined. So this media is chemically defined. This is actually a media that I grew, or I made up in the lab when I was doing my master's degree, growing cyanobacteria. And we know each of the ingredients that we're adding. We know the chemical formulas for them. We know what they are. We know how much we're adding. So we know everything that's in there. Okay, is this media complex or chemically defined? So this is going to be complex. And what was the giveaway there for you in the ingredients list or giveaways? So the most obvious one is the yeast extract, right? So you took, so they grew up a bunch of yeast and then they dried it out and it's this powder and you add it and it's food that, it's nutrition that the bacteria can use. Tryptone is also an extract of proteins as well, but that big one is yeast, the most obvious one. Okay. So is this media complex or defined? So this is TSA agar. If you ever take 222, you'll probably use TSA at some point. So this is also complex. So what was the clue there for you that it was complex? So pancreatic digest, or if you ever see the word digest is kind of in that same camp as extract. So pancreatic digest, papaic digest, both of those are going to suggest that it's complex media and not defined. Yeah. Okay, so now I want you to tell me whether the scenario that I've listed is involving selective media, differential media, both, or neither, based just on the information that I'm giving you here. So you're doing something involving nutrient agar. Is it selective, differential, neither, both, just based on what you know or what I'm telling you? And I know this stuff is tricky. The unit 2 is hard. I'll give you guys a minute to think about it. So nutrient agar, in case you don't know, is just it's a kind of agar that we use that is that a lot of bacteria can grow on, that has stuff in it bacteria can use. So in this scenario, it's not acting as either selective or differential. It's neither. And that's because we're not specifically selecting for or against any certain types of bacteria. And we're also not telling you that some bacteria will look different when they're grown on nutrient agar than others based on the characteristics of the media. So this scenario, you'd say it was neither. Yeah, it is tricky. It's definitely tricky. Okay, so this next scenario, media with penicillin added to it. Penicillin is an antibiotic. So selective, differential, neither, both. So this one, yes, this one would be selective because we are selecting for microbes that are penicillin resistant and against microbes that are susceptible to penicillin. So anything that is susceptible to penicillin will not be able to grow on this media. It will die. But things that are penicillin resistant, you'll see that growth. Okay, so you have media with bile salts to inhibit growth of gram-positive bacteria. Selective, differential, neither, both. So this would also be selective because we are selecting against gram-positive bacteria and for gram-negative bacteria. Yeah, so again, the key is that some stuff is, because of what we'd added, some stuff is not going to grow on it. So this next one, lactose fermenters show up as red, non-fermenters show up as yellow. So selective, differential. So this would be differential, right? Because based on the characteristics of the microbe and stuff we've added to the media, some microbes are going to look different than others based on what they can do. So this last one, blood agar looking for zones of hemolysis. So in this scenario, again, it would be differential because bacteria with the ability to lyse red blood cells will look different than bacteria that cannot lyse red blood cells. Sometimes we use blood agar and we're not really trying to use it as a differential test. We're just trying to grow microbes on it. But when we're using it and specifically looking at the hemolysis activity of the bacteria, then it's acting as a differential media because bacteria that can lyse open the red blood cells will look different than those that cannot. But we didn't necessarily add anything to the blood agar that is going to select against the growth of some microbes and for the growth of others. Okay, these are tricky. That's why we're practicing them. They're hard. Okay, a few more practice questions. Bacteria that require oxygen to survive. So what would their, based on those oxygen classifications, they be obligate aerobes, obligate anaerobes, aerotolerant, facultative, anaerobes? So they would be an obligate aerobe if they require oxygen to survive, yes. And bacteria that can't survive in the presence of oxygen, like Clostridium tetani, Clostridium botulinum, would be obligate anaerobes, right? The temperature classification of most pathogens. So we had psychrophile, mesophile, thermophile, hyperthermophile. So most pathogens are going to be mesophiles because they like our temperature. Ways to culture viruses. What are the methods that we use there that we kind of talked about? There were kind of three. There were the embryonated eggs, right? Which is why there are some vaccines, if you have egg allergies, there's issues for you getting them because a lot of vaccines, they grow in eggs. So animals and cell cultures. Yep, those are the three. Do we have any direct counting methods for viruses? No, none. Because they're too small and they need a host. Indirect counting methods for viruses, there was just one that we talked about, were the cell plaques, right? Those clear areas, the larger the clear area, the more virus you have. And the growth phase where cell growth equals cell death. So there's lag, log, stationary, and death or decline. So stationary is going to be where cell growth equals cell death. The death or decline phase will be where cell growth is smaller than the cell death rate. Okay, that's what I have for tonight. Any other questions or concerns about anything? And I'll have your disease presentation, not your disease presentation, your case study groups posted tomorrow night, so you can see those. And again, make sure you watch the disease presentations before you take your lesson assessment. The disease presentation questions there are 2 disease presentation questions at the end. You'll have those every week, you'll notice that they ask, there's kind of some rotating questions, but they're always asking the same ones. They're always asking you, what's the name of the microbe? Is it bacterial, viral? And then it will ask you things like, what's the reservoir, transmission, treatment and prevention, signs and symptoms. And those questions are worth 2 points apiece, so you'll want to make sure that you don't run out of time before you answer those because they are worth a little bit more. And Angel, if you want to know what you missed on the unit assessment, pop me an email and I'll let you know what you missed. And kind of be watching as you take the lesson assessment that we'll be talking about microbiology concepts, or you'll be learning about microbiology concepts during the week. And then a lot of times the quizzes will want you to kind of apply the concepts that you're learning to the microbes that we kind of talked about in the disease presentations. And so maybe things like, for example, when we talk about antibiotics in lesson 9, vancomycin is really good for gram-positive bacteria, but it's not great for gram-negative bacteria. And so it might ask you something like, the disease presentation bacterium for the week, would vancomycin work on this? And you should be able to say, well, yes or no, based on kind of what you knew about that organism and what you know about that concept. So anyway, kind of be thinking about it that way. If you see a question you don't know the answer, don't freak out. Kind of try and see if you can kind of pull it apart. Okay. And then, no, I didn't. I made up the recipe for your masters. Heather says, so is that why we can't grow most bacteria? So one big thing is that we don't, so 99% of bacteria we can't grow. So one of the things is that we don't know their exact nutritional requirements, right? If they're going to be really finicky about the things that they need, and we don't know those. That's one reason. Another thing is that if you think about it, most bacteria live in communities. They don't live in pure cultures. And there's a lot of interactions and symbiosis that are going on between bacteria. And so it could be that certain bacteria don't really, that in order to get them to grow in pure culture, you would have to figure out what they were getting from other bacteria that might be living in their communities, what nutrients some other bacteria might be giving them, or chemical signals, or different things like that. And so there's that community aspect as well. In addition to not knowing nutritional requirements or other environmental things, what types of community interactions are going on is a really big one as well. So anyway, any other questions? Okay, well, thank you for coming. Hope you guys have a good week.